Welcome everyone, and thank you for joining us for our presentation titled Image and Data Processing for HCS Toxicology. It's my pleasure to introduce you to Dr. David Egan. David is a CEO and co-founder of the Core Life Analytics. And joining David today is Liz Rubitsky. And Liz is a senior scientist in genetic toxicology at Vitro. For a complete biography of our speakers, please visit the tab at the top of your screen. Now, if any questions arise during the presentation, we want to encourage you to submit them in the Q&A box. Our speaker will address your questions following the end of the presentation. Please join me now in welcoming David and Liz. I will now turn the presentation over to them. Welcome to both of you.
Hello there, I'm David Egan, and it's a pleasure to be able to talk to you today about how high-content screening technologies are used for toxicology. Now, to get started, what I'd like to do is just to give you, a brief overview of the history of how we've ended up where we are today. So one of the things that's very important to know is that in the 80s and 90s, there was a large push towards the industrialization of drug discovery. And so there were a number of enabling technologies that came together to allow for this. One was the availability of modern molecular biology and biochemistry techniques, also and genome and gene sequencing. And this allowed people to really get, do a lot more advanced studies on disease processes and allowed for the identification of new drug targets. It also allowed for the development of very specific assays, biochemical assays, for these drug targets. This, when combined with laboratory automation and combinatorial chemistry that allowed for the generation of large numbers of different chemical structures, allowed the development of what became known as targeted drug discovery, where pharmaceutical companies would be able to generate large libraries of thousands or even millions of compounds that they could then test against very specific targeted assays in high throughput and ultra high throughput screens. And the whole idea was that if we could test more targets, we would get more leads, and then we'd be able to generate more drugs. At this stage, safety and toxicology was generally tested here in the preclinical development stage. So if you look at this kind of drug discovery pipeline from the early stage discovery through to the different clinical trials, here just before the clinical trials was where safety was actually studied and compounds were tested for safety. Generally, this was done in animal studies. Now, there was a problem with this that became apparent, whole process that became apparent over the course of the '80s, and there was actually almost a small cottage industry developed in publishing papers about this problem, is that the whole process did not give the expected return on investment. The number of compounds that were actually going to market did not increase in comparison to the number of programs that were being generated and the number of compounds that were going into clinical trials. One of the major problems that people discovered was that a lot of the compounds that were going into clinical trials were failing. There were a number of reasons for this: efficacy, toxicity in animals, but unfortunately also adverse reactions in humans in compounds that were getting through to the market. Also pharmacokinetics, the bioavailability of compounds, and some problems with that. As I mentioned, there were some very high-profile market withdrawals where compounds actually went through these trials, and then you got problems in humans. And what was found that in the 25 years to 1999, about 0.7 approved drugs were withdrawn per year, but in 1994 to 2006, this actually went up to 2.1 approved drugs withdrawn from the market per year. And 80% of these withdrawals were due to hepatotoxicity, liver toxicity, or cardiotoxicity. So if we look at this figure here, this shows what was the scale of the problem. Apart from infectious diseases where you had reasonable success rates, for these other types of drugs in cardiovascular disease, cancer, or CNS disorders, you had very low success rates in phase 1 and phase 2, and slightly higher rates in phase 3. So this is a serious problem. And when you looked at the reasons for compounds failing, yeah, toxicity in animals and adverse reactions in humans had a substantial substantial reasons for the failure. One of the major problems is that, as I said, the compounds at this point were generally being tested in animal studies here at this point for toxicity. But especially with hepatotoxicity, it turned out that only 50% of hepatotoxic drugs would have been detected in animals. So it turns out that animal species do not predict human metabolism of drugs very well, and animal species do not predict the diversity of human patients. Also, normal, healthy animals are not predictive of the effects of drugs and the problems of drugs in diseased human patients. And it seemed that when it came to hepatotoxicity especially, there was a subpopulation of human patients who were particularly sensitive to adverse drug reactions. Other problems with animal studies, they're extremely expensive. Also, over the course of the 90s and the 80s, there became a lot of popular opposition to animal studies. So pharmaceutical companies really came under pressure to reduce the number of animals they were using. Also, from a practical standpoint, animal studies just can't support the higher throughput of compounds that was coming through the pipelines. What was decided is that what we needed in the industry was to identify toxic compounds earlier in the drug discovery pathway. And the idea was that we really needed predictive assays that would allow people to predict whether compounds would be toxic, or and that then these problems could be engineered out with medicinal chemistry, or the compounds could be just rejected, a kind of a fail early approach. And so what this required was in vitro assays, these predictive in vitro assays that could be deployed in a high throughput fashion early on in the drug discovery, because here you're talking about larger numbers of chemicals to be screened, not just a handful of chemicals that you might be putting into animal studies farther down the pipeline. Now, fortunately, there was a successful precedent for this type of strategy in pharmacokinetics. And one of the major problems that had been discovered is that compounds, especially oral compounds, were failing due to a lack of bioavailability. They were not being absorbed in the gut. And so what was developed was an in vitro assay in order to test whether compounds would be absorbed. And this is called the CACO2 assay. And basically, these CACO2 cells, which were I believe, a colon carcinoma cell line, could be plated on a membrane in this little transwell insert here, and they would form tight junctions, so form a tight barrier, so that the only way drugs could get from this compartment to this compartment would be by actually passing through the cells. And this was found to mimic bioavailability via gut endothelium quite well. And you can see here how for a lot of these compounds that it was very highly predictive of their ability to be absorbed in the gut. Compounds that do have problems with this assay are ones that are subject to active transport. And so they could not be predicted because they're highly effluxed by drug pumps in the gut. So before we go ahead, I want to talk about some terminology and define some terms that you'll see coming up in some of the examples that I'll show you later on. And these are about assay sensitivity, specificity, other ways of defining the quality of your assay. So assay sensitivity is how well an assay identifies, in our case, toxic compounds. So it's the total number of compounds detected as being toxic over the total number of toxic compounds that are in our test set. Now, specificity is how accurately it detects toxic compounds. So here, this means essentially the number of toxic compounds detected divided by the total compounds detected. And so you could have an assay that's very sensitive. It's very good at detecting toxic compounds, but it may not be specific, in that a lot of the things that it's detecting may be false positives. Obviously, what you want is an assay that's both sensitive and specific. There are other values that are used, such as the positive predictive value, and so this is how well an assay identifies positives. The negative predictive value, how well an assay identifies negatives. Another important and useful one is the balanced accuracy. If you have the accuracy for detecting, let's say, toxic compounds or non-toxic compounds. To get an overall view of the assay, you could take those accuracies for each of those classes and then get the mean of that. And that gives you an idea of how good your assay is at detecting both. So with that in mind, we can move along, and what we can talk about is the actual in vitro assays that were available to people who wanted to do this earlier detection of toxic compounds. These types of assays that were available at the time were mostly based on very late stage toxic events, cell count, apoptosis, or some sort of events that may not possibly be linked to actual toxicity. So one way of measuring cell viability is to measure ATP levels, but these can go up or go down in ways that do not actually affect cell viability. Similarly, various measures for cell metabolism. The problem with these assays that were already there were that they had, low sensitivity. There are many false positives, but they did have many false negatives, but they did have high specificity in that they detected few false positives. And so you can see here some of these assays outlined in this table, and you can see how the various sensitivities here were quite low. The highest would be around 19%, but the specificities were quite good. So things that you were detecting, then yes, they were definitely toxic. So the upshot of this was that these assays were good for detecting highly toxic compounds, but not really good for predicting compounds that were toxic due to, let's say, chronic exposure, things like that, and certainly not good for detecting what are known as idiopathic compounds, so compounds that only affect a small number of people, and it's not really understood why. The proposed solution then to replace these assays, to come up with more predictive assays, was to focus on earlier events in the toxic kind of process, and events that were more linked to the type of toxicity that you were looking at. So for example, for hepatotoxicity, you would focus on things that are important in liver cells. For example, it was known from the understanding of hepatotoxicity that steatosis was a problem. This is the accumulation of fatty acids. Also, cholestasis, the impairment of bile formation, the accumulation of phospholipids. It was known that reactive metabolites could cause problems, and these could be measured. Another very important factor in hepatocyte biology is, the mitochondrial function. The various functions of hepatocytes require a lot of energy, so anything that disrupted mitochondrial member potential would be a problem, and so this could be measured. Also, oxidative stress, the metabolism causes a lot of oxidative stress, and so the measurement of damaging free radicals was useful. Another type of assay that was used were P450 assays. The cytochrome P450s are very important for the metabolism of drugs in the liver, and compounds that would inhibit these could cause problems with, let's say, drug-drug interactions. So if someone was taking two drugs, one drug inhibits a P450 that's metabolizing the other, then the concentration of the other drug could become very high and cause problems. And so these were also adopted. Fortunately, for a lot of these types of assays, there were various fluorescent markers out there that could be used with cellular systems. A bunch of them are outlined here in this table. So biologists had some flexibility in how to build these assays. As regards the cell types that were available, initially a lot of the work was done in HepG2 cells, which were hepatocellular carcinoma cells that seem to be pretty good at mimicking some certain liver functions. People subsequently adopted cell systems that were more physiologically relevant. For example, primary hepatocytes from humans and animals. Also, more recently, iPS-derived hepatocytes. Also, 3D systems, so sort of mini livers, essentially, in various different 3D systems. And the combination of these cell systems and the various fluorescent markers available meant that people could adopt high content analysis. for these assays. Now, I'll give a very brief description of high content analysis because there will be more in-depth discussions of the overall technology available in some of the talks that are coming up subsequently. We'll talk about those later on. But basically, high content analysis is where you set up a cell-based assay in a multi-well plate, and then instead of taking a simple readout with a plate reader, you actually use an automated microscope to image the cells in the well. So that, in combination with various fluorescent markers, then can give you a very useful opportunity where you can do image analysis, and you can use automated image analysis to extract numeric descriptors of the various labeled organelles or the cells themselves. And so what this gives you then is a very rich, essentially morphological profile of what's going on in a well. High content analysis had a number of things to offer when combined with toxicology assays. One is that you could use it for monitoring live cells easily. Single cell monitoring was possible. Also, you can do simultaneous multiparametric monitoring. So you'll see later on how this offers up lots of opportunities for the use of things like machine learning. Also, it allows for iterative measurements over the same well or even the same cell. And so you can follow things in time. The different types of assays that could be combined in these systems were diverse in that you could have biochemical assays, various immunochemical assays, as well as morphological measurements of the cells themselves. The assay is also very amenable to automation using robotics, so that you could actually get high throughput that was required in earlier stages in drug discovery. Another thing you can do, especially compared to animal studies, is lower costs. It can be more reproducible, and it's also can be combined with more complex cellular systems, iPS cell lines, 3D cell lines, and co-cultures. So the reason I started with hepatotoxicity is that that's probably where the most progress has been made in cytotox assays for using high content screening technologies. And I want to show you a few examples now of various different platforms that have been developed. The first one is an assay that was developed back in the mid 2000s by Peter O'Brien and a group from Pfizer at the old Sandwich Pfizer site in the UK. And this was a high content platform, but was based on 4 different dyes. And so TMRM for mitochondrial membrane potential, fluorophore for intracellular calcium, that increases in cells that are stressed, and then host for nuclear and cell number, and TOTO3 for cytoplasmic membrane permeability. So you can see that here on the left, you can see these are cells that have been treated in a control, and then here cells where you're been treated with a hepatotoxic compound. Now, the cell line that they decided to use here were HepG2s, which was a human hepatocarcinoma cell. And so this has some metabolic activities that make it suitable for these type of work. So the way they set up the experiment was to treat the cells with compounds in 12 doses, and the idea was to get to 100 times the Cmax. This is the maximum concentration of a drug that you would initially see in an animal or a human. If that wasn't possible, try and get to 100 micromolar. As positive controls, they use tween for nuclear size and cell permeability, ionomycin, FCCP, which collapses mitochondrial potential. Now, one key thing is they found that pre-incubation with the drugs was essential, and so incubation of the drugs for 3 days seemed to be required. And then the idea was, after pre-incubation with the drugs, add dyes and then image over the course of around 3 hours, taking images every half hour or so in the 4 different channels. The way they would call drugs or compounds as being cytotoxic was to plot dose response curves for the 4 different measurements, and then any drug that gave a greater than 2 times the standard deviation from the mean negative control within that dose was then called as a toxic drug. Here's an example of the data from a myodarone in this platform. And so what you can see here, these are at each concentration, you can see the time course of the imaging over the course of 3 hours, and for the various different labels. And then what's very obvious here is then you can see here with these doses, everything looks pretty much the same. And then around this dose here for the mitochondrial membrane potential, things start to go awry. Then here you can see with the calcium, it starts to increase here around this concentration. With host, you see start to see changes around here in nuclear area. And then here with the total for cell permeability, then around here you start to see increases in cell permeability. So you can see that the different measurements, you start to get responses at different concentrations. So if we were to look at this, it would probably be calcium would be the earliest, the one that's getting where you're seeing an effect at the lowest concentration here. What was very encouraging when this was analyzed is they looked at a panel of 611 compounds with conventional in vitro assays, and as we discussed previously, the specificity of these types of assays was very high, but the sensitivity was very low. So you were not picking up all the hepatotoxic compounds, but the ones you were picking up were very likely to be correct. With this high content assay, however, in a test with 243 compounds, both sensitivity and the specificity were high. Sensitivity of 93% and specificity of 98%. So this was extremely encouraging. The idea that you could run this assay at an early stage in the drug discovery process and then be able to accurately call and predict compounds that would could potentially cause problems later on. So there were a couple of things that setting up this assay highlighted. One was that for certain drugs, pre-incubation with the compound was essential. For certain drugs here on the left, it wasn't required. You could get effects even without pre-incubation of the compound. But with these ones on the right, it really was necessary to get pre-incubation for up to 3 days. So they arrived at a kind of a standard set of conditions where you would do pre-incubation for 3 days. Another thing that was highlighted was that single cell analysis was very useful, especially for looking at, let's say, the sequence of events that were happening, the different kind of things that were being triggered by individual drugs. And so high content analysis, of course, is very good for this in that you can follow single cells using the high content analysis. Another thing that was highlighted was an effect known as hormesis. And hormesis is where you get an initial increase in a value with dose, but then it starts to go back down again. And so if you were to try and plot dose response curves with the normal models for these kind of responses, you're not going to get a curve. And so a normal curve would look like this, and that works fine with the standard methods. But with these types of curves, you have to come up with a better model in order to fit curves for these. And these are consistent responses. You see these reproducibly, and so they actually used a different model in order to take account of these types of responses. And so this sort of things were very important. So when we look at the various different measurements that were being used, of the 4 different measurements, the highest sensitivity one was cell count, and then nuclear area, and then calcium. The least sensitive parameter seemed to be the mitochondrial membrane potential. The next example is a more recent paper from Litzin Liu and Danielle Zink at the A-Star in Singapore. And this is also a hepatotoxicity assay. And there, the problem they were looking at is that at this point, the gold standard cell system for hepatotoxicity are primary human hepatocytes from sourced post-mortem. Obviously, yeah, these are expensive and can be difficult to get, and of course, yeah, there may be some issues with reproducibility. But what's also another problem with these is that you can get de-differentiation of these primary hepatocytes, and then you get a loss of metabolic activity. So this group were looking for a more reproducible system where you did have as much of the metabolic activity possible. And so one cell line they were looking at were HEPA-RG cells, which were probably, they thought of as being more liver-like than HepG2 cells, and that they had more of the metabolic activity, but where you didn't get this dedifferentiation effect. They also looked at different markers. DAPI for DNA, which of course is common with the last example, but then they also looked at REL-A. REL-A is a subunit of NF-kappa B, with the transcription factor, which gets translocated into the nucleus in response to stress. And this, of course, is quite a dramatic phenotype in a high-content assay. And so they used this as a marker, and also F-actin, looked at F-actin, so which is, of course, a good indicator of changes in cell shape and in the cytoskeleton. And so what they first did was just to look at these HEPA-RG cell lines. They just used standard cytotoxic measurements, annexin 5 for looking at apoptosis, and ethidium homodimer 3 for looking at permeability, cell permeability, and then looked at a panel of cytotoxic hepatotoxic compounds and non-toxic compounds to see if they could measure these with this system. And this looked promising, and so then they moved on to constructing their high content assay with the other markers. And so here again they could get responses with known toxic compounds, and the idea was to generate this as a robust assay, again pre-treat the cells for 16 hours, but then to fix them. Instead of doing live cell analysis, they could fix the cells and then image them. And this use of fixed cells actually makes things, in practical terms, easier to run in high throughput, as opposed to a live cell approach. So this is something that actually makes an assay more practical to run with larger numbers of compounds. Now, one thing that was different between this approach and the previous platform that we looked at is that they didn't just take the obvious, a number of obvious measurements. They actually made use of more of the power of high content analysis. Now, one thing about these high content screening instruments and the image analysis software that you can use with them is that you can actually pull out a lot of different measurements, potentially hundreds or even a couple of thousand measurements, depending on the image analysis platform that you use. Instead of taking one measurement for each of our labels, what this group is, they calculated, many different measurements, and then they used this in this clustering method here, using hierarchical clustering to cluster the responses of all the chemicals in the library. So with the hepatotoxic compounds in red and the non-toxic compounds in blue, and then clustered all of these responses against all of the different features. Okay, and so what you can see is that you get clustering here of a lot of the hepatotoxic compounds over in this area over here. And what you can also see is that there are two features here that are REL-A features. You can see them labeled in here. They're from the REL-A channel that are extremely high in signaling for toxic compounds here. You can see these are these kind of purple boxes here that line up with the red guys, and you don't see them over here with the negatives. So what they decided to do next was to actually take this, now that this kind of gave some confidence that this overall platform could separate the toxic from non-toxic compounds, but they wanted to optimize this. And the way that they did this was actually to use a machine learning approach. The machine learning the method they used was support vector machine, and I'm not going to go into all the details about machine learning, but basically machine learning is what's called a supervised method, where you take classes of known toxic and non-toxic compounds, and then you train a model over that, and then that model can then be used to apply to other data sets, and you get essentially a measurement of whether those other wells are giving responses that are similar to toxic or to non-toxic. One of the kind of byproducts of the vector machine learning model, like a lot of these machine learning models, is that it outputs a rank order of all of the features or measurements according to how important they are for your model in order to distinguish between your different classes. And so here what they found was that in the support vector machine, one of these rel A features that came up in the clustering was the most important feature distinguishing between toxic and non-toxic. And here you can see, if we use that for calling hits and for plotting dose response curves, you can see here that using this as an assay gives a very specific assay, but it's not quite sensitive enough. By that, if you go back to our definitions again, it's very specific in that all of the compounds over here that it's calling as toxic compounds are indeed toxic compounds. Okay. But it's not very sensitive in that you can see that it's actually missing quite a few compounds over here. Here with the negatives, you can see that nearly everything is called negative. It is picking up a few negative compounds as being positive. And so if you look here at the numbers for sensitivity and specificity, you're getting a sensitivity of a little over 50%, but the specificity is quite high. So you're not picking up all the toxic compounds, but the ones that you are picking up are more highly likely to be actually toxic. Okay, and then here's our balanced accuracy. It's out here a little over 70%. This top feature and the idea of taking a single feature and using it for our calling, yes, it could be useful, but there was a possibility to come up with something better. And one thing you can do to come up with a better model is actually to come up with a smaller set of features that do contribute together to give you a more robust assay. And so this is what they did. If we look at if we look at this single feature here, it gives these sensitivities and specificities and balanced accuracies. But what they did was they went back and used what's known as recursive feature elimination with support vector machine in order to come up with a better set of measurements that would allow a better assay. And so the way you do this is that you build and test a support vector machine model, and then you start eliminating the least important features in an iterative manner, and then build and test your model again until you come up with a model that actually gives the best performance. And so in this case, they narrowed it down to a 30 feature set that could be used in support vector machine that actually gave very high sensitivity this time, very high specificity, and a better balanced accuracy. And so this is a nice example of how you can really make use of the full power of high content analysis. You're calculating many features, some of which you may not really understand what their connection is with your biology, but by using machine learning, you can combine these features and come up with a feature set that actually gives you a very sensitive, specific, and accurate model. Okay, we're back to nephrotoxicity again with this platform that was generated by Joachim Gehrig at the University of Heidelberg in Germany. When you're looking at assays, there's always, especially high throughput assays, there's always a trade-off between the practicality and the suitability for high throughput applications and physiological relevance. As we discussed earlier, using whole animals and mammals, mice, whatever, is not practical for very high throughput applications. But as you get down to using more reductionist systems, individual cell lines in single cultures, then you start to lose the complexity of being in the whole animal, and because of that, there may be things that you miss. Fortunately, there are other animal models that are amenable to high throughput and screening, and one of those is zebrafish. Zebrafish have a fairly simple embryo and that can be labeled with GFP, And because zebrafish embryos are transparent, you can then easily image them. In this assay, I think this is one of the is probably the coolest thing I'll show you today. What you can do is you can isolate synchronized zebrafish embryos and plate them in these 12-well plates. In these 12-well plates, they can be treated with candidate compounds that you're testing for nephrotoxicity. And then what you do is you remove the chorion, and what you do then is you put them in a 96-well plate, but in this case there's agarose in the bottom with a channel in it, and then the zebrafish, each zebrafish embryo can fit into a channel, and now the zebrafish embryo they're all in the same orientation. Once you have them in the 96-well plate, then it's pretty straightforward to image them, and with some cool image analysis methods, you can actually crop the image to get down to just an image of the zebrafish kidney itself. And here's an example of a 96-well plate of zebrafish kidneys. And in row F down here, you can see that there is a toxic compound, a nephrotoxic compound, and it's easy to see even by the naked eye that these kidneys are something's not quite right. After this, you have to move on to extract some sort of numeric features from these images. Now, in this case, it's not so easy. You need some sort of manual intervention. And in this study, this group used two different methods. Now, the first one was where a scientist went in and labeled these critical points in each of the kidney images. Then software could easily calculate these distances and angles, okay, to come up with 6 different features that could then be used for output from the assay. Also, there were kind of non-renal features that could be extracted that described various other aspects of the whole embryo. So this was one method that they used. Another method was to build a system where an expert could quickly look at each of the kidneys and score them according to various metrics. So what they did then is they took these different methods and they compared them. And so you can see it here in the clustering where they compared the quantitative method, where the software generated numbers, and the qualitative method where the expert generated the scores for each of the kidneys. And they actually came up and agreed very well. So here you can see the various different nephrotoxic compounds that were detected in both experiments. Now that you have all of these individual scores, what you can also do is you can take those, and those are now a profile that you can use to classify different toxic compounds. Here we see an example of a negative compound, and then in red some positive compounds. And what you can see is that some of the different compounds have quite different profiles. And so this one here, for example, has a very different profile from this one, but this one is kind of similar to this one, maybe more similar to this one. And so this kind of shows you that you can probably get some sort of insight into mechanism of action from the shape of these profiles. So what the group did then is they actually took all of these metrics and carried out principal component analysis. And then this is, of course, a data reduction method that we'll talk about more later. But then what you can do is once you've reduced it down to 3 principal components, what you can do is you can plot the various different points in 3-dimensional space. And here what you can see is that these different compounds, here is ketoprofen, and out here is phenbufen. And you can see that they're in different areas in 3-dimensional space, and this is indicative of the different kind of mechanism of action. Okay. So if you remember back to that DNA relay F-actin assay, we saw that you could use the same assay for toxicity in hepatocytes or toxicity in kidney cells. And even though it was the same assay, we could see that different features were actually most important for the different types of toxicity. So this is kind of interesting how you could use one kind of assay for different types of biology. So that kind of leads to the idea of, okay, well, what if you had an even broader assay that could be even more broadly useful, that could maybe even be completely independent of different types of toxicities or different types of targets. And actually there is something like this, and it's called cell painting. And if you've already been involved at all in high content analysis, there's a very good chance you've already heard about this. So the idea behind cell painting is exactly that, a very rich assay with a lot of different labels that can be used to profile all sorts of bioactive molecules. So here's the idea, is that you can label cells with 5 different dyes. And so this is a very simple assay to run, and that they're all chemical dyes and very useful for automation and live cell imaging. And the dyes are for host for the nucleus, and then the endoplasmic, they label the endoplasmic reticulum, the nucleoli and cytoplasmic RNA, actin, Golgi in the plasma membrane, and mitochondria. So here are the various dyes that are used in the classic cell painting protocol. And the idea is that you can then set up this assay and use it to test various different chemicals or genetic perturbations. Again, go through the standard high content analysis pipeline with automated imaging and then automated image analysis, except this time, because you have so many different labels, and the group ran it through an image. It was developed by the Carpenter lab at the Broad Institute. They ran it through Cell Profiler, which is an open source image analysis platform that group had also developed. And this generates a lot of different features, lots of different texture features for all the different channels, as well as the usual intensities and things like this. And so then you end up with maybe 1,500 different features that give you a very rich data set from which to work with and to generate profiles. Obviously, this could be very useful also for profiling toxic molecules. And so this is exactly what Josh Harrell at the EPA, who's published a couple of different papers on the use of cell painting for toxicology now. And he has a quote about this in a recent Nature webcast, which I would definitely recommend that you check out. And the idea is that, to use cell painting as a very broad kind of assay, which could capture all sorts of different molecular and phenotypic responses. And of course, their interest is to identify compounds that could be toxic in the environment. What Josh and his group did was they actually used cell painting in order to see if they can identify environmentally problematic compounds. So they actually set up 2 different assays. The first one was a cell viability assay. And this one is a more traditional assay where you label cells with host and then use propionimidine to identify dead cells, because propionimidine is not normally cannot normally penetrate live cells. And so this was one assay, and then the other assay was the cell painting assay, where they set up 4 of the dyes, and then use this in their high content experiment, and then generated, not using Cell Profiler but using PerkinElmer's Columbus, 1,300 features. Okay. So now what I need to do is go through a little terminology that they use in this paper. So they have a thing called the benchmark response. And the benchmark response is the response that essentially creates a hit for a particular measurement. Now, for the cell viability assay, this was set around 3 standard deviations from the negative control. For the cell profiler assay, this was set for plus or minus 1 standard deviation from the negative control. Now, a benchmark concentration is for the particular assay and for a particular measurement, that's the concentration at which you get a benchmark response. And you'll see that, for example, here, that is graphed here on the x-axis, the benchmark concentration, and then here are the different measurements. So another key point here is that the measurement from the particular assay that gives you the lowest benchmark concentration is thought of as the point of departure. And then if we're looking at comparing the 2 different assays, whichever assay gives the lowest point of departure is called the high throughput phenotypic profiling point of departure. And that's the point of departure for that particular chemical. Here are some examples, the various different channels that were being used, and with compounds that give strong phenotypes in those particular channels. And then here we see some non-toxic compounds and then some toxic compounds. These are classic toxic compounds, and the responses that they give, it's hard to see here, but this is actually in increasing dose from here to here for each of the compounds. And then these are all the 1300 features for the cell painting assay. And then here are some responses in cell count and cytotoxicity. And then all the features are grouped here according to DNA, RNA, the 5 different channels. Okay. Now this explains how they group the various different features in order to come up with the benchmark concentrations and the point of departure. So this is a little bit complicated, but what you can see here is, here for berberine chloride, these are the responses of all of the features, and these are the benchmark concentrations. So here you can see at this concentration, you're getting, for this particular feature, a very high response. And then here you have features that you're getting no response even at high concentration. So these are the ones that are active. Okay. Now, what you do then is you take all of these 1300 features and you group them into 49 different classes. And so here you can see how they're actually grouped. So for example, you will have DNA intensity features, DNA texture features, DNA morphology features, RNA, etc., etc., etc. And this builds up into, and for the different cellular compartments, it builds up into 49 different groups of features. Categories of features. And then for each category, you get an aggregate score for the actual benchmark response. And then for each one, you get a benchmark concentration. And then the one that has the lowest benchmark concentration, that determines the pod from the cell painting assay. And then you can compare that with the results from the cell viability assay, and then come up with your HTPP pod, point of departure. And so essentially this is what it's telling you, is that what concentration are you starting to get some sort of effect from a chemical, either through the cell viability assay or the cell painting assay. And then compounds that have a very low point of departure are things that you probably should be worried about. As usual, pre-incubation is an issue. And so you can see here that with a lot of these, with compounds, you don't get any response at 3 hours, but then here with this compound, you start to see a response at 6 hours, 12 hours, and then it maxes out at 48 hours. So the general protocol seems to be that it's a good idea to pre-incubate again for 12 hours. The assay is also very reproducible, and so here there are a number of different plates where you're looking at the various different responses, the cell viability propydiamidide response for staurosporin. You can see that they're very consistent across all the different plates. The cell count from the cell viability assay, again very consistent, and here you can see the responses of the cell painting assay, again across all these plates. So across from plate to plate, it seems to be very, very consistent. What was also interesting is that you saw consistent responses across different cell lines, which was also very interesting. So if we think about the tools that you need to do some of this work, well, as regards the image acquisition, you know, all the large instrument manufacturers have automated microscopes, and there are also many smaller manufacturers now who are producing automated microscopes. When it comes to image analysis, not every manufacturer, especially the small ones, have their own image analysis platform, but the major ones do. The likes of Cytiva, Thermo, and Molecular Devices, PerkinElmer, etc. Apart from the commercial platforms that come with those instruments, you can also use something like ImageJ or Cell Profiler. So Cell Profiler is a very well established open source image analysis platform that is very suitable for a lot of this work. As regards image management, well, again, some of the commercial platforms have image databases and data management databases that they package with their instruments. Another option for an open source image analysis database is the Omero platform, which is another, like Cell Profiler, it's a very successful open source tool. As regards to the data analytics for the processing of the numeric data after you've done your feature extraction, well, when you look at these various papers and you get excited about doing that analysis, often you'll find that a lot of the work is done with, coding in things like R and Python, and naturally you'll need someone who can actually do that to help you replicate some of that work. But you can also do things that are very similar to this using some other commercial and open source platforms. For example, with our tool Stratominer, you can do phenotypic analysis and build machine learning models. Similarly, in Spotfire, which is marketed by PerkinElmer for these types of applications. Cell Profiler Analyst also has some machine learning and other analytical tools built in. And then there is a pipelining tool, NIME, which is, it's kind of between coding in R and tools like Spotfire and Stratominer, and allows you to build pipelines using various modules and do image analysis. As regards direct image classification, some of this type of functionality is built into some of the image analysis tools, such as PerkinElmer's Columbus, where you could identify cells and train models based on those. Also, Cytiva have a tool called PhenotLifts that works with their Encarta. These are based on machine learning of the numeric features. There are also some deep learning tools out there, which we think will be very useful for some of this work. At the moment, I think PerkinElmer have something for that, and also GeneData have a tool called Imagence, which is a deep learning classifier. There are also some other open source things like Advanced Cell Classifier is a free tool that is also available. Now, if you look at this in a kind of more general term, then the overall workflow is taking this big data, doing advanced analysis using computation tools, and then using data-driven modeling to kind of identify and classify these various different events, from this kind of molecular initiating event at the molecular level, then out to morphological events in cells, and then maybe later on, using more things like digital pathology, you could look at things in tissues or organs and things like this, so that you can actually build models possibly for each of these different events and try to get a bigger picture of what's going on and how and to be able to intervene to solve these problems as you're going along. Where can the high content analysis methods be useful? Well, definitely at the very earlier stages, as we've shown, they can be very useful of the discovery process. They can be very useful for the predictive toxicology. Later on, also, they can give you very useful information when it comes to working out mechanisms of toxicity, which could be very useful for future programs. And when you look at the overall impact of this kind of work, yeah, there's a possibility that it could really have, when implemented at various stages and using various different technologies, it could have an enormous impact on your decision making as you are working through an actual pipeline. So as to the future, well, one thing that's very obvious is that if you look at our high content analysis, we're looking at one particular type of data there. There's a lot of other types of data that could be generated from various other types of analysis, various different omics technology, metabolomics perhaps, genomics, RNA-seq, various systems biology methods, and I think the future is how to integrate these and pull them together so that you can get a bigger picture of what is going on in the toxicology that you're seeing. So one thing you will notice is during the presentation, I didn't go into a lot of details about high content analysis itself and a lot of the background and how to do it and things like that. the reason for that is because this is actually just one of a series of webinars, and a lot of that information, basic concepts on high throughput screening and high content analysis will be dealt in some of these webinars that are coming up. So there'll be one here by Josh Harrell, who was mentioned during my presentation, and also Mark Anthony Bray, who was involved in the development of cell painting. He'll be giving one, and then later on there'll be another one on 3D 3D models for drug discovery. So I would highly recommend that you get on over to the Labroots website and register for these ones, because together all of these will give you a very nice package of information on high content analysis for various different applications. And so finally, I'd like to thank SBI2 for the chance to help out with this. It's always extremely enjoyable to work with SBI2. You should definitely check out the SBI2 website and have a look at the annual meeting. Hopefully next year that we'll be able to do that live again. Also, I'd like to thank Victor Wong at Core Life Analytics, who was an enormous help in pulling together all the information for this, and Liz Rubitsky, who was also very helpful. Liz works in safety at drug safety at Pfizer, and she was great help in helping us to figure out what we should be presenting here. And Liz should also be available, hopefully, to help out with some of your answers to some of the questions. And so with that, I'd like to thank you all for joining, and we'll be taking some of your questions.
Thank you, David, and thank you, Liz, for that outstanding presentation. And we will now move to the live Q&A portion of our webinar. As a reminder, please submit your questions in the Q&A box. So let's get started. We have a couple of questions already here from our wonderful audience. Our first question is, could deep learning be useful for toxicology?
Yeah, I'll take this one. Yes, that's a very good question, and unfortunately, it's a topic that I had hoped to cover, but there was just so much other cool stuff. I thought maybe we'll leave that for another webinar. Deep learning, of course, is very useful for image-based screens, and there are a number of publications out there that have used that. Another way to use it is actually not on the images themselves, but deep learning with the structures and combining structural information and calculated parameters from structures with previous tox data. And so this could be used to build models that then could be applied to new compounds. So there was actually I know in 2016 there was an Austrian group that built a tool called DeepTox, and that was used that approach. And of course there are actually now commercial tools out there also that are using deep learning for images. I know PerkinElmer has built something for Columbus. Cytiva have a thing called Synap that they recently launched, and Gene Data, as I mentioned at the talk, Gene Data have a thing called Imagens. So these are things that people could possibly use. Yep. One challenge with deep learning, of course, is the amount of data that's required to build good models. So that is one challenge for that.
Wonderful. Thank you so much, David. Our next question is, what do you see as the biggest hurdle for applying this approach to 3D co-cultural models, specifically spheroids models?
Liz, do you have any thoughts on this one?
Boy, so we have an investigative tox group that does our 3D spheroids, and I believe that the biggest hurdle is just consistent cell growth, figuring out what size spheroid you need as far as the wet lab work. And as far as the data, I think, as David mentioned before, the sheer amount of data is probably a hurdle. David, did you have any other thoughts as far as the data analytics?
Yeah, I mean, so the number of objects that you want to acquire, that's going to impinge on the amount of data that you have for, let's say, machine learning and things like this. If you're just taking, let's say, organoids as objects, but then of course when you get to the volume of imaging data, if you're doing Z-stacks, that of course is going to be a huge amount of data. So yeah, that would be a major challenge. As technically, I guess one of the biggest challenges would be actually imaging within 3D structures. And of course, this isn't just for tox, but this is for any type of 3D structures. If you have larger structures, can you image deep into it to get good quality data? So yeah, so I think it's the volume of data and then the standard kind of how do you image in 3D structures. Yep.
Thank you so much to both of you. And I want to thank our audience again for these great questions and remind them that any questions that were submitted and not answered today will be answered via their email. We have time for one more question. How should one deal with high autofluorescence in primary hepatocytes?
Whoa. Liz?
Yeah, so we've run into autofluorescence issues before, and I will say with the imager that we have, we are able to actually do some manipulation of the image, which I know is a bit controversial, but we are able to do that to get rid of some of the autofluorescence. The other thing that we do is we use a far red dyes as often as we can. So again, I think it mostly comes down to your imager and what options you have available.
Yeah, that's a good one.
Thank you so much. And I want to thank both of you for your time today. And I also want to provide you with a little moment to provide some closing remarks for our audience. I'll start with you, David.
Yep. So just in closing, I realize this is not a hugely comprehensive look at high content analysis for tox. I'm sure that there's lots more stuff out there. But the idea is to give people a taste of what's possible. And, at another time, maybe we can get into things like deep learning. One thing I would kind of reiterate is please have a look at the other talks, especially for those who, don't have a big understanding of a lot of the background of HCA. Some of the other talks will kind of fill that in. We just weren't able to get them in the right order because of scheduling conflicts. And so definitely Josh Harrell and Mark Anthony Bray's talks will be really cool for filling in those gaps.
And Liz, did you want to add anything?
Just that I think that we're in a great point for starting to really see advances with high content in toxicology. It's just going to keep getting better, and it's an exciting time to be in it. The machines, the technology, and the reagents that are coming out are great. I would highly recommend attending user group meetings for the various companies. It's a great way to get to understand the company and what's available. And to meet other people and ask questions there.
I want to thank you both again for this wonderful presentation, and I want to thank our audience members and let them know that this presentation will be available for on-demand viewing in the SBI2 virtual conference for 12 months. So please remember to share it with your colleagues who might be interested in today's topic. And do not miss out on the other presentations on our agenda. Thank you again for participating. Until next time, stay safe, stay healthy, and take care. Bye-bye, everyone.
Thank you, Susie. Bye-bye.
Bye-bye.